Yes, okay. Now is my favorite slide. Why do you think that is? Guess what? We get to talk about cell injury, okay, hypoxia, alright, and necrosis all on a molecular level. What? I know it. Awesome, right? Isn't that sweet? I'm excited. And I swear I'm not a nerd either. Alright, so we're going to start off by asking the question, why does a cell even need oxygen at all? What's the point of oxygen? So, here's a cell membrane here, and I'm going to ask you, what ion is most prevalent on the outside of the cell? Sodium. What about on the inside? Potassium. And it's actually very important for a cell to maintain this equilibrium, and we'll see why in a little bit. But just trust me, it's important. So, what pump then maintains this equilibrium? I wonder if it's the sodium potassium pump. Wow. Whoever named that is a genius. Okay. So this pump actually exchanges how many sodiums? 3 sodiums from the inside for 2 potassiums. And it requires energy in the form of ATP. As does almost everything that requires energy in the cell. We saw it earlier with apoptosis, and you'll see it in just about every other reaction within the cell. Okay, so ATP is required. Now, where do we get ATP from? So to answer that, we have to talk about your favorite subject of all, biochemistry. I know. You're probably shutting your eyes already and snoring, but no, biochemistry is fun, and I'll show you why. I promise it's going to be fun. Once we tie all this stuff together, you're going to be so happy that we did. Okay, so here's a little biochemistry snapshot, and we're going to actually have to talk about, the electron transport chain. You thought you were done with this. You thought you put it away back in first year med school. Nope, it's back. And back with a vengeance. So, the electron transport chain transports electrons. Where do these electrons come from? They actually come from NADH. Where does NADH get its electrons? Well, from glycolysis and the TCA or Krebs cycle. Remember all those dehydrogenase enzymes? Yeah, those all make NADHs. Alright, so NADH deposits its electrons onto the electron transport chain, and when it does that, it actually converts itself into NAD+, because it gets oxidized. So, electrons jump from complex 1 to complex 3 to complex 4, and then to another substance that we'll mention in a little bit. But each of these complexes is progressively more electronegative and more possessive of electrons, until the final electron acceptor, what is that? Oxygen! That's why we need oxygen. Oxygen takes these electrons, complexes with them, and turns into water. Alright, so what's the big deal with the electron transport chain? Why do we need electrons to jump around? So the reason actually is that each time an electron jumps from one complex to another, it pumps out a proton into the intermembranous space of the mitochondria. So these protons go into the intermembranous space, and this actually sets up an electrochemical gradient because of all these protons in this space. These protons really would like to flow down their gradient. And if they do, this will actually provide enough potential energy. The drop in potential energy will actually be enough to phosphorylate something. So they flow through only one little gap. Only one little gap allows the protons to flow, and that is ATP synthase. And each time a proton flows down its electrochemical gradient, it actually phosphorylates an ADP molecule into ATP. Woohoo! So that's good news. That's how we make ATP, and that's why we need oxygen. So, ATP phosphorylates the pump, sodium is pumped out of the cell, and then potassium is shunted inwards. Very cool, right? Very awesome animations too, if I might add. Alright, so now let's talk about what happens when we don't have oxygen. Uh-oh. So this is called hypoxia. Alright. And here we have ATP. So why won't we have ATP anymore? Well, let's again bring up this biochemistry snapshot to explain why. So no oxygen. Remember, there's nothing to accept electrons now. Oh no. So if these electrons that were from NADH can no longer flow through the complexes and be accepted by oxygen, then there will be stasis of electrons. No flow will mean no pumping of protons into the intermembranous space, and thus no flowing of protons down their gradient and phosphorylation of ADP to create ATP. Wow. Everything finally comes together. You see how beautiful it is? I think it is. But that is why we need oxygen to accept electrons. Otherwise, no ATP. Alright, so now let's talk about the downstream effects of this. So, there's no ATP, then there's nothing to phosphorylate this pump. If this pump isn't phosphorylated, sodium can't leave the cell. If sodium can't leave the cell, what always follows sodium? Here is why we see that this pump is so important. What follows sodium? Water. And water will rush into the cell and cause it to swell up. And that's the first main change of cell injury. It's still reversible. We'll talk about the reversible changes first, but it's not very good. So, the cell will swell up because the sodium can't leave, and even some of the organelles within the cell will start to swell. Like the rough ER will swell, ribosomes will detach. Then what do ribosomes do? They make protein, right? So you will lose the ability to make proteins. I mean, that's no good. And what type of metabolism do you think the cell will start to undergo? Actually, we'll start to perform glycolysis. That's not very good either. Because without any oxygen around, that's the only way it can make ATP. How many ATP per glucose molecule do you think it'll make? About 2. Whereas with aerobic respiration, it was making 38. So 2 is only a fraction of the amount that it could make with oxygen. That's terrible. But it's all it can do, so it's just working with what it's got. All right. Another change we see is that the cell starts to turn pink or becomes hyalinized. And why is this? It's because of a drop in pH or acidification of the cell. And why does this happen? Well, guess what? To explain this, we have to talk about biochemistry again. What? You don't really mind? You sure? You actually kind of like biochemistry now? Wow. Okay. Well, we'll talk about it then some more. So, again, a drop in pH will actually cause the cell to turn pink, but also the nucleus to clump up like that. But here's the biochemistry snapshot. So, remember what I said, that there's no more flow of electrons anymore because there's no electron acceptor, so there's a stasis of electrons in these complexes. So that means that NADH won't be able to dump off any more electrons, and there will be a really large buildup in NADH and a decrease in NAD+. Now the problem with this is that NAD+ is actually required for glycolysis, some of those dehydrogenase reactions, and glycolysis, we need that to make our measly 2 ATP. Shoot. So we need to regenerate NAD+. Even though it's not very helpful, it's all we've got to make ATP. How are we going to do that? How is the cell going to do that? So to answer that, what is the final product of glycolysis? Pyruvate. Okay. And guess what? When pyruvate actually gets converted into something, NAD+ is regenerated. What does it get converted into? Lactic acid or lactate. The problem with lactic acid is that it's an acid. Oh no. So that's why we get the drop in pH. So now it all makes sense. So we get NAD+ and we keep making ATP, but at the cost of acidifying the cell. Alright, there's another reaction that actually regenerates NAD+. And do you know what that is? It's actually turning dihydroxyacetone phosphate, well, that's a mouthful, into glycerol 3-phosphate. So glycerol is actually the backbone of triacylglycerides. Believe it or not, When you string 3 fatty acids onto glycerol, you have yourself a triacylglyceride. And that's why there's a buildup in fat within the cell. So these are all of the reversible changes so far. Now we start getting into the irreversible changes. Time to salute our poor little cell goodbye. The first major irreversible change is a buildup in calcium. So just like there's a sodium-potassium ATPase pump, there's also a calcium ATPase pump that requires ATP. Normally pumps out calcium. It can't work anymore. Calcium, therefore, builds up. What's the problem with that? Calcium is a potent stimulator of all kinds of degradative enzymes within the cell. Oh no, it's just going to cause so much more destruction and damage to this poor cell. So enzymes that will tear up the membrane and the nuclear membrane and organelle membranes, like even lysosomal membranes. The contents leak out and destroy things. That's just no good at all. So the nucleus, like I said, will be degraded, and there will be, what's this called, like fragmenting of the nucleus, karyorrhexis, and there will be karyolysis, further destruction of the nucleus, and eventually nuclear pycnosis, where it just shrinks down into nothing, basically. Oh, that's so sad. Oh man, I feel for the cell. Eventually, some of the organelles, like I said, will lose their membranes. If the mitochondria loses its membrane, what's within that gets released? Cytochrome C, remember that from apoptosis? Yeah, that'll go and activate caspases, which will then go degrade even more things. So just everything's going to get destroyed. Eventually, once the cell loses its phospholipid bilayer, its membrane, that is the last straw. The cell is toast when all of its contents just leak out. Such a sad tale. So that's why you've got to take your oxygen, kids.